Johari, your biochemistry mentor. In this video, we are going to discuss a very important, clinically important topic, Sphingolipidoses. So let's start up with our discussion, Sphingolipidoses. Now, if we look at the word itself, the word is self-explanatory. What is sphingolipidosis? It is simply accumulation of a sphingolipid. Right? Accumulation. Now, the question is why a sphingolipid will accumulate? And what is a sphingolipid? So, first of all, we have to see that what is a sphingolipid and what happens when it is accumulated and why it is accumulated. Accumulation is always, remember, in biochemistry, I have told you that if I say that A plus B is forming C, right? Once the function is over, C will be converted into A plus B. But the question is, say, if that enzyme which is required to break is absent, it will lead to accumulation of that particular substance. So, I can say that if there is a deficiency of the enzyme leading to the degradation of sphingolipid is absent, it will lead to accumulation of sphingolipids. Now, the question is that which organelle is responsible for the degradation. So, in general, we say that lysosomes are the ones which are responsible for that. So, first of all, sphingolipidosis is they are the lysosomal storage disorders. Very, very important. Storage disorders. Now, the question is, they are the lysosomal disorders and it occurs due to the deficiency of the hydrolysis group of enzymes. Lysis enzyme. Right. Now coming over to this, what is this sphingolipid? Now, in my previous video, in my earlier videos, which I have actually, they are exclusively on the phospholipids and glycolipids, I told you that the name itself is self-explanatory. If I say sphingophospholipid, it's clear phosphate is there. If I say sphingolipid, no phosphate is there. Okay, so first of all, let's just try to visualize what is the actual difference between this. So, sphingolipid first of all. If we dissect it, it will be dissected into sphingosine and lipid hai, so obviously it is an ester. Now the question is, what is sphingosine? This is a unsaturated alcohol which has 18 carbon atoms. So I can say C18 and it is an unsaturated alcohol. Right. And if we talk about the structure, it has this type of structure. C18, it has OH here, it has NH2 here and it has OH here. Now, when this amino group is attached to a fatty acid, this becomes a ceramide. So, we can say ester has come. Okay. Now, the second is, if this ceramide is attached to a carbohydrate, it will become a glycolipid. Right. So, if I say what is a glycolipid? Glycolipid is the one which is formed of, say, if I talk about glycolipids, they are formed of what? Sphingosine, 'sphingosine' 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 fatty acid, right, which is a ceramide, which is further linked to either glucose or it is linked to galactose. Okay, so if it is linked to glucose, it will be a glucocerebroside. So I can say glucocerebroside will be there. If I talk about galactose, so it will be a galactocerebroside. Right. So always remember what is the basic difference between glucocerebroside and galactocerebroside. Obviously, glucose is there, sugars are different. But if we talk about their occurrence, glucocerebroside is always present in, not in CNS, very important, only in extraneural tissues. Okay. So, glucocerebrosides are never present in central nervous system, but yes, they are a part of extraneural tissues. And if we talk about galactocerebrosides, they are always present in CNS. Okay. So, we can say these are what? These are the cerebrosides. Now, coming over to, we know that glycolipids are of two types, cerebrosides and gangliosides. Okay. Now, if we talk about, ye to ho gaya, these are the cerebrosides. If we have to talk about the gangliosides, what will be the structure of a ganglioside? Simple, sphingosine will combine with a fatty acid plus, instead of glucose and galactose, they will either have oligosaccharides or they will have NANA, N-A-N-A. So, this becomes a ceramide, right, and this becomes a globoside or a ganglioside, and this becomes a cerebroside. So, now you have understood what is the difference. This is first, this is second. And NANA stands for N-acetyl-neuraminic acid. Minic acid. Clear? Because why I am telling you the difference? Because if any of the enzyme degrading these are absent, there will be accumulation of that particular sphingolipid, and that is why they are known as Sphingolipidoses. And what will happen wherever they are present, it will lead to enlargement of that particular organ. So, in general, if we talk about hepatosplenomegaly is the most common one in Sphingolipidoses. There will be inclusions, cytoplasmic inclusions, or the cells will be laden with lipids, either cerebrosides or gangliosides. So, first of all, let's see what is the difference between cerebrosides and gangliosides. So, if we talk about the difference between cerebrosides and ganglioside, we can simply say, as the name suggests, that cerebrosides are ceramide linked with glucose and galactose. That is what just now we studied. Ceramides linked with either glucose or galactose. If glucose, glucocerebroside, galactose, galactocerebroside. And when ceramide is linked to NANA, means some derivatives, oligosaccharides, जैसे N-acetyl-galactosamine, N-acetyl-glucosamine, N-acetyl-neuraminic acid, then they will become gangliosides. Then cerebrosides occur only in the white matter of brain and the myelin sheath. Gangliosides in the grey matter and obviously ganglions, gangliosides. Then types are glucocerebrosides and galactocerebrosides. And here they are globiosides or gangliosides, GM1, GM2, GM3, GM4. And the functions, we know conductions are, it conducts nerve impulse and they are the biogenic amines. Related disorders we are going to study later, that's why I have hidden this because that is what we are going to discuss now. So this is the basic difference between cerebrocytes and gangliocytes. Okay. Now let's go over to this schematic diagram, that how they are synthesized, and if something is absent, it leads to which disease. So this is a very interesting thematic plan. We will start with sphingosine. Right. Now, if we start with sphingosine, I told you when a fatty acid comes, it becomes what? It becomes a ceramide. That is what we have studied just now. Okay. Then this ceramide, if it combines with a base which is choline, and obviously phosphate is also there here, it becomes what? Sphingophospholipid, or it is known as sphingomyelin. Right? So it becomes sphingomyelin. Right. Now, if this ceramide combines with glucose or galactose, what it becomes? It becomes a glucocerebroside. Or it becomes a galactocerebroside. Or I can simply say that they will become cerebrosides. Right? Now, these cerebrosides, when they combine with oligosaccharides, then they will become globosides. And these globosides, when they combine with NANA or some derivatives, N-acetylneuramic acid, NANA, they become the gangliosides. So this is how the chain goes. Ki sphingosine, we started with the parent compound sphingosine, which is just an unsaturated alcohol. When it combines with a fatty acid, it forms a ceramide. This ceramide, when it combines with a phosphate and a base, it becomes sphingomyelin, which is lining the myelin sheath. If it combines with glucose or galactose, it forms glucocerebroside or galactocerebroside. If it combines with oligosaccharides, they form the globosides. And if it combines with NANA, it forms the gangliosides. So this is how the thematic plan goes. Now we are talking about the sphingolipidoses. Sphingolipidoses will be when, say, this enzyme is absent, when this enzyme is absent, when this enzyme is absent, or this enzyme is absent. So it will lead to accumulation of that particular thing. Right? So if I say if beta-glucocerebrosidase enzyme is absent, there will be accumulation of glucocerebroside. Beta-galactocerebrosidase, there will be accumulation of beta-galactocerebroside. Similarly, oligosaccharides, again accumulation of this. So this is how the chain will go. Okay. So now we are going to study, first of all, the most important, or you can say the most common lysosomal storage sphingolipidosis, Gaucher's disease. Then after that, later on, I will just draw the table. So I will just write A here, B here. Okay, first of all, let's take A. So A is Gaucher's disease. Right. Now, if we talk about Gaucher's disease, the deficient enzyme, just see the thematic plan. I can simply say that glucocerebroside is the one which is forming. So, the deficient enzyme is beta-glucocerebrosidase. Okay, now what are the clinical features and basically what happens in Gaucher's disease? First of all, it is the most common lysosomal storage disorder. Right? Deficient enzyme is this. So it leads to what? The most important is, if you talk about the occurrence, I told you it is present only in extraneural tissues. It is not in CNS. So the most important one is no mental retardation will be there. But since there is enlargement, because there is deposition of these glucocerebrosides, so what will happen is it will lead to hepatosplenomegaly. Right? There will be thrombocytopenia. Very, very important. Thrombocytopenia, which will lead to actually the person, there will be reduction in the RBCs count. So all the time the person suffering from Gaucher's disease will have a kind of fatigue or weakness. Okay, then there will be bony pains also. And obviously if hepatosplenomegaly is there, there will always be a discomfort. Right, it is going to disturb the appetite also. And the most important, thrombocytopenia is there, and there will be inclusion of these cerebrosides, glucocerebrosides, which will form a type of, cytoplasm will be known as, it will be like Gaucher cells laden with this glucocerebrosides. So cytoplasmic inclusions will be there. Plasmic inclusions. I'll show you the images also. So they are actually known as Gaucher cells. Right. And another important feature here will be, if we take the X-ray of the femur में क्या हो जाएगा? Due to the deposition, it gives a Erlenmeyer flask type of structure. So it will be Erlenmeyer. It is actually known as Erlenmeyer flask deformity. Right. So femur will be looking like this. The structure of femur will be like this. Okay, and what are the other features? I'll just show you the Gaucher cells first of all. You can see that this will be the Gaucher cells. They are known as Gaucher cells. And it gives a wrinkled type of appearance. So that is why these Gaucher cells are also known as, they are crumpled or tissue paper like appearance. Crumpled tissue paper appearance. Right. And another picture, a very interesting picture about these Gaucher cells is this also, this Gaucher's disease. You can see that this is a very interesting picture. See, there will be enlarged spleen and liver. Due to enlarged liver and hepatomegaly, I told you, it will lead to abdominal swelling and tenderness will be there. Okay, thrombocytopenia, low red blood cell count, it will lead to fatigue. Low platelet count, it will lead to easy bruising and bleeding, and reduced bone density will lead to bony pain and the fractures. And the most important one is femur will have a, Erlenmeyer type of deformity. So this is Gaucher's disease. The main thing is the deficient enzyme here is beta-glucocerebrosidase. Right, and that is why if you look at the picture also, I have marked it as A. So glucocerebroside, or it is Gaucher's. Second is B. Ab second में galactocerebrosides, so it is like alpha as well as beta. So इसमें we will take first of all beta. And yes, one more thing, if we talk about the treatment in Gaucher's disease, treatment is enzyme replacement therapy is available. Okay, enzyme replacement therapy is available. Now, next we come over to the B. Up in B, I have written one is alpha, other is beta. So, first of all, we will take up the two diseases. One is Krabbe's disease. Right? And second, related to this is Fabry's disease. Now, if we talk about Krabbe's disease, the deficient enzyme here is beta-galactosidase. And if we talk about Fabry's disease, the deficient enzyme here is alpha-galactosidase. Now, what is the difference between Krabbe's disease and Fabry's disease? The most important point to remember about this is this Fabry's disease is the only X-linked sphingolipidosis. Sphingolipidosis. Right. And if we talk about the inheritance of this is autosomal recessive. In fact, all are autosomal recessive except Fabry's. Recessive. Okay, now if we talk about the main feature which distinguishes Krabbe's disease from rest of the sphingolipidosis is that here no hepatosplenomegaly will be there. If we talk about Krabbe's disease, no hepatomegaly. Okay, and if we talk about Fabry's disease में there are so many Fabry's means F stands for febrile episodes of fever will be there. This is sometimes it will be like high, sometimes it will be low, sometimes it's like abrupt. Febrile episodes of the fever will come into this. And A stands for angiokeratomas. There will be formation of, like hardening of this thing will be there. Then hypohidrosis. The individual who is suffering from, the male actually because it is X-linked, hypohidrosis, diminished sweating will be there. And he will always have an urge to put his hands in water. Okay, so Fabry's disease mein the most important I would say clinical features would be, the most important is hypohidrosis, diminished sweating, febrile episodes of fever, right? Then R stands for kidneys, so kidneys also affected. And Y say you can say because only X-linked na, so Y chromosome is present in only males, so just like it's like a mnemonic also to remember. And angiokeratomas. Right. So these are the main features of a male suffering from Fabry's disease where the deficient enzyme is alpha-galactosidase or alpha-galactocerebrosidase. And in Krabbe's disease, if we see, it is autosomal recessive and no hepatomegaly is observed in this. Whereas rest of the features, if we talk about this, mental retardation will also be there. Developmental delay will be also there. Alright, so these are the symptoms of a person suffering from Krabbe's. Then moving on to If we look at again the thematic plan comes, this is, chalo I'll write C here. Okay, now what happens here is, here it leads to Sandhoff's disease. Sandhoff's disease. And here it will lead to Tay-Sachs disease, which we will just discuss now. Okay, and here it will be Niemann-Pick disease. Pick disease. Okay. So I can say C, this will be D, and this will be E. So सबसे पहले C and D we are going to take together. So if we talk about the C and D, C is Sandhoff's disease. Right. And here if we talk about the deficient enzyme here, the deficient enzyme here is hexosaminidase A and B. Again, if we talk about the mode of inheritance, it is autosomal recessive. Always remember, all are autosomal recessive except Fabry's disease. This you have to keep in mind. Fabry's, with the mnemonic is like Y. So Y is, you can say XY. Immediately you can just remember the genotype of a male. Y hai, so it is X-linked. Rest of them are autosomal recessive. Now, if we talk about Sandhoff's disease, the main clinical features here are, now this hexosaminidase enzyme, ye kya hai? This is having two subunits, A and B, alpha and beta. So, if alpha is absent, only Tay-Sachs disease is there. But if alpha and beta both are absent, it leads to Sandhoff's disease. And if we talk about the clinical features, the most important feature is that there is cherry red spot on the macula. This is very, very important. Then you can say cardiac abnormalities will be also there. Right? There will be bony pains. And the child who is suffering from this will have, it's like whenever we just put that rattle in front of him, he will be scared. So he will be totally startled by any sound which makes. So it's like, there will be neural delays also. Okay. Motor delays will be also there. So we can say motor delays will be there, or you can say developmental delay will also be there. Hyperacusis will be there. Startled by the sound of the rattle also. So, matlab the child will not be able to hear anything. He will be totally baffled up. Okay. And Tay-Sachs also resembles Sandhoff's because the deficient enzyme there is hexosaminidase A. But the basic difference between Tay-Sachs disease and Sandhoff's is, in Tay-Sachs no cardiac abnormality is there. Rest of the features will be similar. Okay. I'll see if there is any picture on Tay-Sachs disease, so I'll share it with you. See, this is the formation of that. You can see that. This is the cherry red spot. It's clearly visible, which later stages might lead to blindness also. Lead to blindness. Okay, so this is Sandhoff's disease, right? And again, if we talk about an accumulation is what? Accumulation of globocytes is there. Because if you see again the thematic plan, globocytes, again I will just write it here because see cerebrocytes are there, no? Cerebrocyte, when oligosaccharide comes, it is forming globocytes. So there will be accumulation of these. So accumulation of globocytes. And these globocytes, when they combine with NANA and acetyl-neuraminic acid, then they form gangliocytes. And gangliocytes may, again you have to just remember, we are not going into that much detail, but yes, here if there is absence of enzyme hexosaminidase A, it will lead to Tay-Sachs disease. So another disease is Tay-Sachs disease. Right? And if we talk about Tay-Sachs disease, the deficient enzyme here is hexosaminidase A. Aminidase. And what happens here is there is accumulation of GM2 gangliocytes. So there is a difference between Sandhoff's and Tay-Sachs. Tay-Sachs may again accumulation is there, but of these GM1, GM2, so here it is GM2 gangliocytes. Okay, whereas in Sandhoff's disease, accumulation of globocytes is there. So here Sandhoff's disease is there, and here Tay-Sachs disease is there. And if we talk about the clinical features again of Tay-Sachs disease, the clinical features will be like same, it will be there. Hyperacusis will be there. The most important one is cherry red spot formation. But no cardiac abnormality is there. Abnormality is there in Tay-Sachs. Right? So this differentiates Tay-Sachs from Sandhoff's disease. So this is Gaucher's, Krabbe's, Fabry's and Sandhoff's along with Tay-Sachs. Now one more disease is there. Again, if I go to the chart, Sphingomyelinase or Niemann-Pick disease is there. Okay, and this is also there. So here this is E. When there is accumulation or when there is absence of enzyme ceramidase, it leads to Fabry's disease. So you can see that in this schematic plan, all the different type of Sphingolipidoses are there. Okay? So now we are going to discuss what happens in Niemann-Pick disease and what happens in Fabry's disease, or I will just write it as F. Okay, A is clear, B is clear, C is clear, D is clear. Then we come over to E. See, this is D. Again same cherry red formation is there, so you can just relate that because this is asked as an image based question also. Then we come over to E, which is Niemann-Pick disease. Now Niemann-Pick disease may, if we talk about the deficient enzyme, the deficient enzyme is sphingomyelinase. Right. So obviously sphingomyelin. Ab just go recall that what is this sphingomyelin. So if we talk about the composition of sphingomyelin, it has sphingosine, which is connected with the fatty acid, which is connected with phosphate, and which is connected with choline, which is the nitrogenous base. Abhi we were discussing about that. Usme there was no phosphate, there was no nitrogenous base. But here phosphate is also there and a nitrogenous base is also there. So this is your ceramide and this is sphingophospholipid or sphingomyelin, you can say, or it is sphingophospholipid in general. Now when this sphingomyelinase is absent, there is accumulation of this and it leads to basically developmental delay is also there. If we talk about the clinical features, the most important one is zebra body inclusions are there. Bodies inclusions. Just share the picture how this looks like. So there is, because most of the students get actually confused between Gaucher's disease and this. Gaucher's disease में वो जो cytoplasmic cell उसमें tissue paper type appearance, that wrinkled paper appearance is there if you observe clearly in the under the microscope. और यहाँ पे it is like this. So we call them as zebra body inclusions. And rest, if we talk about the features, every feature will be similar. What is there in you can say Sphingolipidoses? All the mental retardation will be there because there will be deposition of this in the myelin sheath. So obviously there will be delay in the nerve conduction also. So it will lead to developmental delay. Clear? Again cherry red spot formation will also be there. So all the features will be similar, but the only feature which can distinguish this with that is that zebra body inclusions will be there. Okay, and these will be actually due to the formation of these ceramides. So this is what? This is Niemann-Pick disease. And then we come over to the next one, Fabry's disease. Now in this Fabry's disease, the deficient enzyme is 𝗵𝗵𝗵𝗵𝗵, as you saw in that diagram also. So the deficient enzyme is 𝗵𝗵𝗵𝗵𝗵. And Fabry's disease mein basically what happens is there is accumulation of ceramides in the joints. So what happens is there is restricted movement of the joints, obviously, because if it is accumulated it is difficult. So it mimics rheumatoid arthritis, but it is totally different from rheumatoid arthritis that we already know. So यहाँ पे I will just write that clinical features are it mimics rheumatoid arthritis. Mimics. Right. So obviously the movement will be restricted and you can see that like this. Okay? There is swelling. Wherever the joint is there, there will be a swelling. So this mimics rheumatoid arthritis. So this is what is Fabry's disease. Okay. So these are the different type of Sphingolipidoses. Now one more disease is covered in this. Halki it is not a Sphingolipidosis. Okay. It is not a lysosomal disorder also, but it is associated with lipids, which is Wolman's disease. Right? So, in Wolman's disease, if we talk about the deficient enzyme here is acid lipase. Right, or there is deposition of cholesterol esters. Right. But generally we consider this under this. So I would say it is not a Sphingolipidosis. Okay. So here what happens is the deficient enzyme is acid lipase or there is deposition of cholesterol esters. So what are the clinical features here? The most important one here is that relentless vomiting will be there. Relentless vomiting will be there. uncontrolled vomiting. Watery green diarrhea will be there. Green diarrhea. And it will be difficult for the individual to survive. So, failure to thrive. And the most important feature is calcification of the adrenals. Calcification of adrenals. I'll just show you the picture also. It's the most important pathognomic feature of this. You can see this. Calcification of adrenals is the most important feature of Wolman's disease. Okay, so these are the different type of Sphingolipidoses. Now let's summarize what all you have to remember in this. Yeah, pehle I will just write the main points, that all Sphingolipidoses, this is very important, all Sphingolipidoses are autosomal recessive. Somal recessive except Fabry's disease, because we know Fabry's disease is X-linked, right? Second important point is all Sphingolipidoses are having mental retardation as the main feature, except Gaucher's disease. Right. Then all Sphingolipidoses have cherry red spot on the macula except Gaucher's disease and Krabbe's disease. Even though in Gaucher's B it has been seen, but Gaucher's A is the most common one, so Gaucher's A में no cherry red spot formation is there. And the fourth important point, all Sphingolipidoses have hepatosplenomegaly, except Tay-Sachs and Krabbe's disease. Very, very important. Krabbe's disease. Right. So, these are the points by which we can actually differentiate between the different type of Sphingolipidoses. All Sphingolipidoses are autosomal recessive except Fabry's disease, which is X-linked. All have mental retardation except Gaucher's disease, because I told you Gaucher's disease mein the deficient enzyme is beta-glucocerebrosidase, which is not there in CNS. Okay. Then all Sphingolipidoses have cherry red spot on the macula except Gaucher's and Krabbe's disease. And all Sphingolipidoses have hepatosplenomegaly except Tay-Sachs, because always remember one more point, that gangliosides because Tay-Sachs is due to accumulation of GM2 gangliosides, So all gangliosides are not present in the liver. Right? So this is also a very, very important point. So and if we talk about the bodies here, so zebra body inclusions are in this, that also I will write. Zebra body inclusions, characteristic feature of sphingomyelinase or Niemann-Pick disease. Right? If we talk about globocytes, or you can say globosoil ceramides, it is in Sandhoff's disease. And if we talk about the cytoplasmic or crumpled tissue paper, crumpled tissue, or the Gaucher cells, it is Gaucher's disease. Right? So, if we quickly summarize that what are the different type of sphingolipidoses, this table you need to learn by heart, because which is very, very important. So, if we talk about the name of the disease here, and if we talk about the deficient enzyme here, we'll start up with the first one, Gaucher's disease, the most common one. So, if we talk about Gaucher's, the deficient enzyme is beta-glucocerebrosidase. If we talk about Krabbe's disease, right, and there is a difference between Krabbe's and Fabry's disease. This is just a mnemonic to remember. This Krabbe's disease is due to the deficiency of beta-galactosidase or cerebrosidase, and Fabry's is due to alpha-galactosidase. And Fabry's is only X-linked. Rest, this is autosomal recessive, this is autosomal recessive. Okay, then after Fabry's comes Tay-Sachs disease and Sandhoff's, which are more or less parallel. So, Tay-Sachs and Sandhoff's disease. Tay-Sachs में the deficient enzyme is hexosaminidase A. Aminidase A. Whereas in Sandhoff's, it is hexosaminidase A and B. Both. And then comes Farber's disease. Farber's disease में the deficient enzyme is ceramidase. And last but not the least is Niemann-Pick disease. And Niemann-Pick disease में the deficient enzyme is sphingomyelinase. Right. Now I am not writing Wolman's disease here because Wolman's disease, chalo, फिर भी I will write Wolman's. But Wolman's may always remember beta, it is due to the deficiency of acid lipase, which is not a sphingolipidosis, acid lipase. So, these are the different, all the sphingolipidoses, very, very important. Right? So, this is all about the video on sphingolipidosis, which is a class of lysosomal storage disorders. How they occur? They occur due to the deficiency of the hydrolysis enzyme. The hydrolysis enzymes which break these sphingolipids into their original components. And always remember sphingosine, because most of the students get confused between sphingosine and glycerol. Both are different. Sphingosine is an unsaturated alcohol which has only 18 carbon atoms. And when it is combined to a fatty acid, it forms a ceramide. Okay. So if we have to differentiate between, first of all, we have to move like this, जैसे phospholipids or glycolipids are there. If we talk about phospholipids, phosphate has to be present. Then the alcohol may vary. So if the alcohol is glycerol, we will call it as glycerophospholipid. If the alcohol is sphingosine, it will be sphingophospholipid, but phosphate will always be attached. Okay. But if we talk about simple glycolipids, glycolipids may glycerol will be never present, phosphate will not be present, only the alcohol which is present is sphingosine. This sphingosine will combine with a fatty acid forming ceramide, which is further attached to either glucose or galactose or oligosaccharides or nana. But no phosphate will be there. And this is how you have to move in a chronological order, then you will never confuse, because majority of the questions have been asked on the structures also sometimes. These means what are the subunits of this? Because sphingolipidosis clinically is very, very important. And always remember, all are autosomal recessive except Fabry's. Fabry's mein I told you mnemonic kaise yaad rakhna hai? Y. Y means XY is a genotype of the male. So only Fabry's disease is X-linked, rest of them are autosomal recessive. So that's all about the video on sphingolipidosis. Thank you so much.